So the question is, what is special about the neuron that allows it to travel, to convey a signal from point A to point B? And to understand that question, you need to understand something about membrane potential and electricity, ions, and how they move inside and outside a cell. So you can't understand the nervous system and biology really without understanding biochemistry and chemistry, right? We've talked about that so many times at the beginning. So this yellow thing here is representing my cell. And around it, you'll notice I have a bunch of ions. Some are positive, some are negative. I got negative ions on the inside, I got positive ions on the inside, I got positive and negative on the outside as well. And your typical cells have well, you tell me. So let's just take a look what's happening. There is a mixture of positive and negative ions, both inside and outside the cell. What the cell will do is it will spend energy to establish a concentration gradient. And what makes a neuron special is that it can change its membrane potential. Most cells, it will just remain the same, but a neuron can change it. So in order for a cell to establish a membrane potential, it uses a very special pump, something we talked about in biochemistry, and that was the sodium-potassium pump. So I have this area here representing the outside of the cell, this here representing the inside of the cell. And what happens is the sodium-potassium pump, it establishes a concentration gradient of sodium and potassium ions on either side. The sodium-potassium pump, it will establish this membrane potential by pumping out 3 sodium and by pumping in 2 potassium. In biochemistry, once upon a time, we learned about the antiport nature of this particular pump. This requires energy. The cell is actively using energy to establish this potential. And you're thinking, okay, why? Why would the cell use all this energy to do this? We'll get to that. The sodium-potassium pump is always on. So it's always pumping out 3 and always pumping in 2. When there's all this sodium on the outside, it's going to try to come back. And there's some channels inside the membrane that allow a little bit of leakage of the sodium back in. We're not really happy about this. This just happens. And there's a little bit of leakage of potassiums leaking out. So the membrane is not completely sodium proof and not completely potassium proof. And so as long as the sodium potassium pump's working, it will establish a huge concentration of sodium on the outside and a huge concentration of potassium on the inside. Okay? So if we measure it like we would measure anything, we use a small little millivolt, microvolt meter, and we are able to measure this, and we'll find that the inside of the cell, because of the movement of more positives out and less positives in, it means that the overall cell charge will be negative. The membrane polarity of the cell is negative. It's a negative value. All right, so this negative value, it can change. So a common negative value for the resting membrane potential for the cell, common value is negative 70. Depending on what resource you look at, you'll notice that it ranges, but I'm going to use a value of negative 70. So in this diagram here, what I have is the inside of the cell here, the outside of the cell here. I have these blue circles representing my sodium ions, and these green circles representing my potassium. If there is a trigger, so these channels here, these green represent potassium channels, and these blue represent sodium channels. And this diamond is my sodium-potassium pump. So the sodium-potassium pump, what it's doing is it's pumping out 3 sodium, and it's pumping in 2 potassium at the same time. So it's establishing this potential. Now these sodium, they want to come back, and some of them will leak across, but for the most part, they can't get across. But let's say, an event happens, and in that event, there is a sodium channel that gets opened, because these sodium channels, they're sensitive to events. So some kind of stimulus occurs, and it opens up. So when it opens up, it will cause some sodium to come in. But if the event is really small, then what will happen is the sodium channel will just close right back up again. Okay, and then the sodium-potassium pump will grab those sodium and pump them back out. Not a big deal. But if the event is large enough, if there's some kind of trigger that's large enough, and it opens up this channel, and enough sodium comes in, then what will happen is the sodium will stream in, and as it streams in, it causes the membrane polarity to go from -70 to a number that's not as negative, right? Because I'm bringing in these positive ions. So I'm going to go from -70 to -65 to -60 to -55. And if we hit the magic number, we'll call it the threshold. If we hit the threshold at that event, at that particular number, then every single channel in that area will open up. When every single channel in that area opens up, then we get this mass influx of sodium come flying in. And as the sodium comes flying in, it causes the membrane potential to rapidly go from the negative value up to 0 and even up to the positives. So these channels here, we call them voltage dependent channels. Depending on the voltage, they'll be open or closed. So at -55, all the sodium channels open up in that area. And as they come in, they cause other parts of the cell membrane that are further down, that are away from these, to also become depolarized, is the word that we use. And it'll cause those channels to open up, and then you get this wave of depolarization that just moves along the cell. Now, it gets less negative, becomes 0, becomes positive, and around a 30, then something interesting happens. These voltage-dependent channels, they shut down, they close. So the sodium channels will close. And around 30, and it's relatively quick, around 30, the potassium channels will open. And they'll just all open, right? So when they open, so what does that mean? It means now these potassiums that are sitting inside the cell, they can leave. And as they leave, the cell becomes less positive, right? Less positive, eventually hitting 0, and it'll become more negative and more negative. And these potassiums will keep on flying out. They'll fly out of the cell, right? And eventually you'll get to a point where it will get, past the threshold, this -55, down to 60, and the potassium channels will begin to close around 60, 65, but they're really slow. They close very slowly. And as they close slowly, what will happen is the whole system will go below the resting membrane potential, and the cell will become hyperpolarized. And during that hyperpolarization event, this cell can no longer be stimulated. It won't accept any stimulus whatsoever. And that's okay, because you got the sodium-potassium pump. What does the sodium-potassium pump do? It's going to pump out 3 sodium, and it's going to pump in 2 potassium, and it's going to keep on doing this until resting membrane potential is reached. And even then, it will still keep on doing it. So this whole event of depolarization, repolarization, hyperpolarization, and now it's stuck in the hyperpolarization event, the sodium-potassium pump never shut down. It was always working. It's just not as fast as these channels. But eventually, when these guys all close, then the sodium-potassium pump will reestablish the membrane potential. That is how the membrane of a neuron works. So let's look at it from this graph perspective. Okay, so I'm actually using -65. That's what I have here on my graph. So let's start off. We'll use red here to represent my graph. So the sodium-potassium pump's doing its thing. It's pumping out sodium, it's pumping in potassium, and the world is right. And then along comes some little trigger that causes a sodium channel to open up, but it was a very weak trigger. So sodium-potassium pump establishes resting membrane potential again, and it's back to normal. But then along comes a stronger stimulus that causes enough sodium to come in for the membrane potential to reach -55. At -55, known as threshold potential, So we're past the resting. So at negative 55, known as threshold potential, at this point, every single sodium channel in that region opens up. And so the cell quickly becomes depolarized, that's the event, passes 0, and around 30 or so, the sodium channels close, potassium channels open. So right here, it's now going to have a whole bunch of potassium leaving the cell, and the cell's going to become less positive, it's going to hit 0, it's going to become more negative, more negative, more negative, more negative. It's going to start closing the potassium channels, but they're super slow to close, so the whole membrane potential drops below the resting membrane potential. Now the sodium channels are all, potassium channels are all closed, and the sodium potassium pump will reestablish slowly but surely the resting membrane potential. And that is the action potential. So a bunch of things just happen. You'll notice I use a bunch of terminology. Let me see if I can change my view here. I'll zoom 150. There we go. So at a depolarization event, that was this, where the cell went from negative 65 to positive 30. Then at repolarization, where it went from positive 30 back down to resting membrane below it. Had hyperpolarization, where it's below resting membrane potential. And those are all y-axis, right? Y-axis. The x-axis is time. So this action potential, once you reach this point right here, until we get back to here, this whole thing is known as the action potential. I used a terrible color, sorry about that. Action potential. It's all or none. When it happens, you can't stop it. It will not stop halfway in between. It just goes. Okay. So the action potential here is measured across the x-axis. And luckily I wrote action potential here, so you can see that. And this event where it is under hyperpolarization, the length of time that is in hyperpolarization is called the refractory period. For you athletes out there, for those of you who are trying to run fast, jump faster, swim faster, the refractory period, there you have a relative refractory period and an absolute refractory period. And this is something that is, it works with you or against you. Some people have the genetics that they have a relatively short refractory period and their muscles twitch super fast. Other people have the genetics where the refractory period is longer and their twitch is a little bit slower. During this refractory period, your cell cannot be stimulated in any way. So it's done until it reaches this resting membrane potential where the voltage gates are now unlocked and ready to be activated again. So each of these action potentials represents every single action in the nervous system. That includes sound. Action potentials are going from your ear to your brain. Vision. Action potentials are going from your eye to your brain. Touch, they're going from your finger to your brain, and so on. They're all just action potentials. Where the action potentials come from, the frequency, all that will determine how you interpret red, orange, yellow, green, loud, soft, melodic, raucous, hot, cold, rough, smooth. They're all action potentials. It's really quite amazing and baffling when you think about it. Okay? So here's a better diagram with everything labeled for you. That's the action potential. So the action potential is something that you are responsible for knowing, and in your worksheet, you'll notice I've given you a number of tables to fill in to help make sure you clarify that concept. So this action potential, what it does is it moves along the neuron. And so we call that movement propagation. So here I've drawn an axon, right? So one part of the neuron. And I'm going to use this spot right here. These are going to be my sodium channels. And so along comes a stimulus. And the stimulus, it's going to cause the sodium channels to open up. And when the sodium channels open up, in comes the sodium. And so the sodium causes that region of the membrane to depolarize. So the membrane now is depolarized. And so what that will do is these sodiums, when they come in, they'll cause the sodium channels that are beside them to also open up, and then they'll allow sodium to come flying in that region, and that region will become depolarized. This region over here, which had, I should have potassium in here to begin with. This region here, now that it's undergone depolarization, the potassium channels will open up and they'll allow potassium to escape, and this whole side here will become repolarized. And so what's going to happen here, I'm just going to get these out of the way because they're kind of in the way. What's going to happen is you're going to get this wave of depolarization. Each time these sodium channels open up, they're going to cause the adjacent ones to open up. But it only goes one way, because once these open up, they'll close and the potassium channels will open up, and you'll get a wave of depolarization that follows. And then the last stage is that hyperpolarization, where you cannot get any activation whatsoever. So this hyperpolarization ensures that the action potential moves in one direction down the axon. This whole concept is known as propagation of the action potential, and you'll see that on the PowerPoint. I'll write the word here, but you'll use the PowerPoint to propagation to get that definition. Propagation of the action potential. The bigger your nerves, the faster these action potentials can move, and the faster conduction can occur. If you are an invertebrate, a squid, you got these big giant nerves. In first year, second year, you guys might experiment on nerves and frog legs or some invertebrate. In our bodies, those nerves are super thin, and the thinner the nerves, the slower. So we need another mechanism that will allow our transmission to occur quickly. And we have it. Remember that myelin I talked about before? The myelin, it will encircle the neuron, and specifically the axon, and it leaves these little spaces in between the myelin cells. These little spaces are called nodes of Ranvier. And so what will happen is the sodium will fly, sodium and the potassium will only come in and out in those nodes. So the sodium will come flying in, and it will cause a depolarization at this spot. And then it won't be able to cause the adjacent cell membrane to depolarize. It actually skips over to the next node. And so we have this thing called saltatory conduction. Saltatory conduction. And so the depolarization event, it jumps. And so in our body, thanks to the myelin sheath, the nervous transmission happens very quickly. Jumps, jump, 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 super fast. So we don't need to have thick nerves because we have the myelin sheath. You lose the myelin sheath, and then your nervous transmission slows down and might stop working entirely. So that's not good. Okay? So that is how the action potential is propagated down the neuron. So now we make it to the end. We make it to the synapse. Okay? So at the end of our axon, we have a synapse. So I'm going to draw it here. And here I'm going to draw a dendrite. So this is the post-synaptic. So this whole area here, I'll do it in yellow because we can't see yellow very well. This whole area here is called a synapse. And a synapse is made up of the presynaptic cell or membrane. It's made up of the postsynaptic membrane. So the action potential is moving along, right? I'll use this red here. It's moving along. We got blue wave, the red wave behind it, and the hyperpolarization thing behind it, right? It's all happening. And so the action potential makes it to here. We're at the end. So now what? How do you get an action potential to go from this presynaptic membrane to here? Because you got this big giant space. Well, it's not giant, but you got a space in between these two axons. How does it make it from here to here? Well, sitting inside the presynaptic membrane are these vesicles. And these vesicles are typically filled with a neurotransmitter. I'm going to use something called acetylcholine, but it could be anything. It could be dopamine. It could be a histamine. It could be epinephrine, right? So I'm going to use acetylcholine. So green here equals acetylcholine, ACH. So as the action potential moves down the presynaptic membrane and makes it to the presynaptic, down the axon and makes it to the presynaptic membrane, it causes voltage-dependent channels here to open up. Up until now, those voltage-dependent channels I was talking about were sodium. But there are other ones as well, and these ones here are calcium-dependent. So these calcium-dependent channels will open up because they become depolarized, and they will allow the influx of calcium. And then through a not a very well-known mechanism, this incoming calcium will cause these vesicles to move. They'll migrate and they'll bind to the presynaptic membrane. And when they bind to the presynaptic membrane, they will release the neurotransmitter into the synaptic cleft. Right? So as these get released in the cleft, what process is going to occur? Well, it's in a region of high concentration here, it's in a region of low concentration over here, so it's going to diffuse across the cleft. So it will diffuse across the cleft, and there's very specific receptors on the postsynaptic membrane for that neurotransmitter. For the acetylcholine, it was histamine, there'd be histamine receptors. It was serotonin, there'd be serotonin receptors. So the acetylcholine will bond to these receptors. Depending on the property of the receptor will cause one of two things to occur. In the case of acetylcholine will cause sodium channels. These receptors will cause sodium channels to open up. And when sodium channels open up, we know exactly what happens. Sodium comes flying in. And as the sodium comes flying in, it causes a depolarization event. And now we have the propagation of an action potential. I'm using red here. I use that for calcium over there, so hopefully you don't get confused. Maybe I'll use a different color. So propagation of the action potential. So it will move, and now the action potential is free to move down this dendrite through to the next axon, and so on. So that's one possibility. This acetylcholine caused a action potential. Because it caused an action potential, this event is called an excitatory postsynaptic potential. You'll see the full word written out on the PowerPoint. So any neurotransmitter, that's what this is. The neurotransmitter is the chemical messenger of the nervous system. Any neurotransmitter that causes a depolarization event, this is called an EPSP, right? So this can cause an action potential. But that's not the only choice. There could be something else. There could be another axon. And it's possible that this axon here has a neurotransmitter inside of it. And when this neurotransmitter gets released, I'll use a different color. I will use, I don't know how many colors I've left. What have I not used? Purple? I've used purple, brown, gray, yellow, orange. So let's say this orange comes in, and let's say this neurotransmitter bonds to a different receptor. Right? And this receptor, instead of opening sodium channels, it opens potassium channels, right? So it opens these potassium channels. So when potassium channels opens, what happens? The potassium leaves, right? And this whole thing will become hyperpolarized, which is the opposite of an action potential, right? So that means if this neurotransmitter causes potassium channels to open up, we end up with something called an IPSP, an inhibitory postsynaptic potential. So think about what could happen here. I could have a dendrite here, and I could have a whole bunch of axons. And let's say that this one here released something that produced, what I had green? I don't remember what color it had. Green, okay. So let's say this one here released acetylcholine that caused an EPSP. And let's say these three over here released another neurotransmitter that caused an IPSP. Right? So this causes an IPSP, this causes an IPSP, this causes an IPSP. So we kind of call this negative, 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 and we call this one here positive. So over here, what's the effect? We have a negative, and we have oops. We have one positive and three negatives. So that means overall, you're going to get no action potential because the one that's going to produce an action potential is overridden by these three that causes three IPSPs. So now you have all these circuits, and the synapses in our brain, they're not made up of 4 or 5, they're made up of hundreds, if not thousands of these at every single synapse. So now you get to see how complex the brain can be in terms of how it talks to itself. Okay, so this is how an action potential is propagated, moves down the axon, comes to the synapse, produces a event that causes the vesicles to bond to the membrane, releasing neurotransmitters that then diffuse across the synaptic cleft, bind to the postsynaptic receptor causing an EPSP or an IPSP, and then we get so many different things that we associate with our brain. Okay? When these EPSPs and IPSPs come, so one single EPSP might not be enough to hit threshold. You might need 2 or 3 at the same time. So if we get one IPSP, clearly that's not going to work. So when you get the right combination of EPSPs and IPSPs at a particular spot, this is known as summation. Right? And summation needs to happen at the right time. So they have to happen at the same time and typically at the right place. So there are 2 types of summations, temporal and spatial summation. So the combination of all these things together is how our nervous system, our brain, is able to sense, perceive, and instruct these EPSPs and IPSPs. Like I said, there's so much information happening here. If you look at the PowerPoint, you'll see more information that talks about the specifics of these that you can copy down into your worksheet. And I'm going to end the section on membrane potential and nervous system mechanics